May virtual journal club. So this is, we're trying to do it monthly and discussing cutting edge topics in the technical field related to data science. Dashu is a 401c3 nonprofit organization. We have quite a bit strategic partner, CAP, SFASA, BAYES. We are a young organization founded about 3 years ago. Membership is free. As long as you register for one of our events, you will receive emails only on our future event. There will never be advertisement, and we never share your email contact with any other organization. The administrative cost is very low. All fundings go to serve the community. We now have over 1,000 members all over the world, mostly in North America, Asia, and Europe. The goal is provide a virtual community for people interested in data science to exchange ideas and help each other. And we organize international scientific conferences. We are actually organizing 2 conferences just this year. One is in early November in Bay Area, focusing on the local biotech pharmaceutical companies and discussing statistical and data science challenges. So look for, just keep tuned in. We'll send you more information when that is more ready. The other one, we're going to do our first international conference in Shanghai in early December. That's our Dashu annual conference focusing on precision medicine and data science, using data to make smart and better choice for physicians. And we're actively seeking members, volunteers, new ideas. If you have any idea you want to know more about us, contribute, sponsor, be a speaker, what have you, just send your email to dashuinfo@gmail.com. Next slide. So we do journal club. We're trying to do it monthly. Before we start talking about today's topic, I just want to bring to your attention, we just finalized the detail this morning, so I didn't get a chance to put it into the slide deck. But we also have our June journal club settled. So the June journal club is Advanced Precision Medicine through Data Science. It will be on June 13th. At 4 p.m. Western time, 7 p.m. Eastern time. We'll send you more about the detailed registration later, but that topic will be presented, this very hot topic, by Rong Cheng, who is the VP of R&D and Bioinformatics at Semaphore Genomics and the Director of Clinical Genomic Informatics and Assistant Professor at H.Han School of Medicine, Mount Sinai. So that's next month. And this month, next slide, please. We are very lucky to have Dr. Han to tell us, again, a very hot topic in pharmacogenomics, the genomic landscape and pharmacogenomic interactions of clock genes in cancer chronotherapy. And Dr. Han has a very impressive resume. He received his PhD from Chinese Academy of Science and continued his postdoc fellowship at Stanford and MD Anderson Cancer Center, 2 of the most prestigious cancer research institutes in the world. And Dr. Han joined UT Health in 2015 and supported by Cancer Prevention Research Institute of Texas recruitment award. And Dr. Han's lab mainly performed computational analysis covering all areas of cancer genomics, focusing on novel genetic elements, such as today's presentation. So without further ado, I will give floor to Dr. Han.
Okay.
And I will mute everyone, but just operational, and then unmute Dr. Han, and then we can reserve most of the questions towards the end, such that, because otherwise there will be a lot of background noise.
Sure. Okay, so I can start. Okay, so first I want to thank you for this great opportunity to present our recent work published on cell systems. The title of my presentation is The Genomic Landscape and the Pharmacogenomic Interactions of Clock Genes in Cancer Chronotherapy. So I listed my email address here. So if you have any questions later, you can. I will be very happy if you can send me the email and we can further discuss. So circadian rhythm is a biological process that displays an oscillation pattern of about 24 hours. These 24-hour rhythms are driven by circadian clock genes, and they have been widely observed in plants and animals. So this figure is downloaded from a Nobel Prize website. You know that the Nobel Prize awarded into the circadian field last year, and this figure shows the basic circadian rhythm for the human body. And we can see that at different time points, the human physiology status will be quite different. You can see that at some certain time point, the human body will have the highest temperature, while in some other time point, they will have the lowest body temperature. Of course, they will also have different blood pressure and they will fall into deep sleep status. So a few years ago, a comprehensive study generated the transcription data across different tissues in the mouse genome. They basically show that there are many, like thousands of genes show the oscillation patterns in different tissues, and they show that this kind of oscillation pattern is largely tissue specific, largely tissue specific pattern, and we can see that in the liver tissue there are more than 3,000 genes show the oscillation pattern, while in some other tissues there are more than about 1,000 genes show the oscillation pattern. But we know that mouse is a little bit different from human because mouse sleep at daytime while humans sleep at night. So a more recent study published in Science I think a few months ago, they studied the transcriptome from different tissues from bamboo. They actually collected 64 different tissues, and then they collected those tissue samples at every other 2 hours over 24 hours. Their results show that more than 80% of the protein coding genes show the oscillation pattern at the transcription level. So you can see that all this oscillation pattern, the color bars. So the mammalian circadian machinery is very complicated and controlled by the transcriptional regulatory feedback loops. And we can see that here that the clock and the BEMA, they will form a complex, and this complex will activate the expression of the clock genes such as the PER and CRY. And on the other hand, the translational product of these clock genes like PER and CRY will coming back to inhibit the activity of this complex. On the other loop, clock and BEMA will also activate the expression of the NR1D family and RR family. So this system including 14 genes are defined as the clock genes. Besides all these clock genes, there are many other clock associated genes. They can also interact with these two transcriptional loops. These genes involved in the circadian rhythms and control the variety of the biological process in a temporal precision. So circadian genes maintain the normal circadian rhythms at the normal condition, but recent study also show that the functional loss of the core circadian genes, such as here the example of the BEMA and PER2, they will induce disruption of the circadian rhythms and accelerate the tumorigenesis. So disruption of the circadian rhythm has been reported that to associate with the cancer development and is also associated with the worst clinical outcome. Therefore, we know that the circadian disruption will lead to cancer. Therefore, there are several of the studies. They developed a way to treat the patient at a different time point, and they define this as a cancer chronotherapy. The cancer chronotherapy is proposed to treat patients at an optimal time, such to increase the drug effects but decrease the toxicity. This figure summarizes all the drugs has already been shown that have the effects in the cancer chronotherapy. For example, this dot represents one drug. If treated the patient at this time point, they can have like more than 150% improvement than treat the same drug but at a worse time point. This dot shows another drug this drug is even stronger if we treat the patient at the best optimal time point. Then it will get like more than 2% improvement than treat at any other time point. So therefore, it's very necessary to develop cancer chronotherapy to improve the treatment effects. So here in this study, we have a lot of large-scale data resources. For example, the Cancer Genome Atlas. The Cancer Genome Atlas TCGA consortium generate a lot of genomic profiling data across like 10,000 patient samples. They covered more than 30 tumor types. On average, for each tumor type, there are on average 500 samples. So TCGA consortium generated the somatic mutations, copy number variations, gene expression, DNA methylation, micron expression, and protein expression based on RPPA. More importantly, they also collected those clinical information such as the stage, age, grade for all those patient samples. These data resources provide us a very good opportunity to understand the mechanism of the tumor genesis. We also include several other large-scale data resources, for example, the Cancer Cell Line Encyclopedia. These data resources, including the transcriptome data for more than 1,000 cancer cell lines, they also include the drug response data. And another two large-scale data resources is the Cancer Therapeutics Response Portal and the Genomics of Drug Sensitivity in Cancer. These two data portals also include a lot of drug response data across different cancer cell lines. So first, with these large-scale data resources, we want to know what is the extent for those clock genes show dysregulation pattern across different cancer types. So in this figure, the x-axis is the different clock genes. The red ones indicate the core clock genes, the 14 core clock genes, while the black ones are the clock-associated genes. The y-axis is different cancer types. And each dot represents the difference between the tumor and the normal samples. The red dots indicate that this clock gene is highly upregulated in the tumor samples, while the blue dots indicate that this gene is downregulated in the tumor samples. And we can see that some of the genes is upregulated in multiple cancer types. For example, this ARNTL2 and AR1D1, they are upregulated in most of the cancer types. In contrast, you can see that many other genes show the downregulation pattern in the tumor samples. For example, the PER family, PER1, PER3, and PER2, they all show the downregulation pattern across multiple cancer types. We also observe that the RR family, like RRA, RRB, and RRC, they show downregulation pattern, as well as the CRY family, the CRY1 and CRY2, also show the downregulation pattern. So this box summarizes how many cancer types show the downregulation or upregulation across different cancer types. We can see that among these 14 core clock genes and 37 clock-associated genes, about 46 of them, more than 90%, show the dysregulation in at least one cancer type. So then we map all those dysregulations into the transcriptional regulatory feedback loop. We can see that for the clock repressors, for example, the CRY family here are downregulated in most of the cancer type. The PER family, including the PER1, PER2, and PER3, also show the downregulation pattern in most of the cancer type. The NR1D1 here shows the upregulation in most of the cancer type, while the NR1D2 show the downregulation in most of the cancer type. For the activators, it shows the very complicated pattern. For example, the ARNTL2 show the upregulation in most of the cancer type, while the clock show the downregulation in two cancer types. The RR family here show the most of the downregulation across different cancer types. Given the large number of alterations for these clock genes across different cancer types, the next question is we want to ask, what is the functional effects for the alterations of circadian genes in cancer? So TCGA provided the RPP protein expression data based on the RPPA, and for this protein expression, we have a way to calculate the pathway score to show that how this pathway score has been signaling pathways has been affected. So here we divided the tumor samples in each tumor type into two groups based on each individual clock genes. So if this gene has clock gene has a high expression and is associated with the high pathway score, then we define this clock gene is associated with the activation of the signaling pathway. In contrast, if these samples with the low expression of the clock genes and these samples show the high signaling pathway score, then we can define this gene is associated with the inhibition of the signaling pathways. So here's the results. And we can see that the alterations of the circadian genes across different cancer types, they definitely lead to alterations of the signaling pathways. For example, the PER2 is associated with inhibition of the apoptosis signaling pathway, also the cell cycle signaling pathway, but they are also associated with the hormone A, hormone B, PI3K, RAS, RTK signaling pathways. In the contrast, the ARNTL2 shows a quite different pattern. The ARNTL2 is associated with the activation of the apoptosis and cell cycle signaling pathway, and also is associated with the inhibition of the hormone A, hormone B signaling pathways. So we know that most of the circadian genes have at least, most of the core clock genes, they have at least one paralog genes. So we want to know what is the functional effects of the paralog circadian genes in cancer. Are they the same or they are different? So here we can see that at least for the signaling pathways, the paralog genes showed quite different pattern. For example, the Cry2, again, they are associated with inhibition of the cell cycle and apoptosis signaling pathway. However, the Cry2 show very few alterations on these two signaling pathways. This suggested that Cry2 may play with the dominant role in cancer, while Cry1 is quite different from the Cry2. So previous studies show that if you knock down the PER1, PER2, or PER3 in the genome, then they will lead to the circadian disruptions. They also show that if you perform the double knockdown of the PER1, PER2, or triple knockdown of the PER1, PER2, PER3, they will show even worse circadian disruptions. So here we want to know what is the combination effects for those circadian genes on the oncogenic pathways. So again, take the Cry1, Cry2 as an example. We further divided those tumor samples into two groups. One group is with the high expression for both Cry1 and Cry2. One group is with the both low expression for Cry1 and Cry2. And we can see that again, Cry2 kind of lead to most alterations in oncogenic pathways, while Cry1, Cry2 has fewer alterations than Cry2, but it has more alterations than Cry1. These results suggested again the Cry2 has the dominant role in cancer, but Cry1 seems to play as a complementary roles to the Cry2. So after we have examined the transcriptional dysregulation for the circadian genes, we want to examine the genetic alterations of these circadian genes across different tumor types. So this is a busy figure. So the x-axis shows the different cancer types. The number of samples in each cancer type are also plotted here. And the y-axis is the different clock genes. The red ones are the core clock genes, and the black ones are the clock-associated genes. The number displayed here is the number of samples with the mutation for this clock gene, and the color define the mutation frequency. We can see that the clock genes generally have very low mutation frequency, suggesting that probably the circadian genes are very important. So there are two genes, the EP300 and CREBBP. They have a relatively high mutation frequency. The EP300 regulates transcription via the chromatin remodeling, and is important in the process of cell proliferation and differentiation. CREBBP is known to play critical roles in the embryonic development. So we can see that most of the clock genes show the relatively low mutation frequency. Here's another display for the core clock genes, and we can see that most of the core clock genes show the mutation frequency less than 2. You can see that all the 2 is here, and most of the circadian genes show the mutations in less than 2 samples across different cancer types. And among all those core clock genes, the PER family, the PER1, PER2, and PER3 has the relative larger mutation frequency. Despite the fact that the circadian genes have a few mutation frequency, they still can have the impact on the clinical survival. For example, in the breast cancer, we can see that those patients with any mutations in the core circadian genes, they have the worst survival. For those samples with the mutations in the PER1, PER2, and PER3, they also have the worst survival. This suggested that even with the low mutation frequency, those clock genes may still affect the patient's survival. So previous studies have already shown that there's a disruption for the circadian rhythms in cancer. Here, we want to examine the extent of the circadian disruptions across different cancer types. So we perform simple calculations. Basically, we calculate the number of genes correlated with each of the individual circadian genes across different cancer types. And we then compare this number with those number of genes correlated with the clock genes in the normal samples. And then we use the number in tumor samples minus the number in normal samples. We can see how many genes lost their correlations to the circadian genes in the tumor samples. Basically, we can see here the x-axis is the tumor type, while the y-axis is all the clock genes. And we can see that most of the clock genes lost their correlations. Like, for example, take this as an example. We see a minus about 2,000 here, suggesting that there are 2,000 genes lost their correlations to the clock genes in the tumor samples compared to normal samples. So for the correlation among those clock genes themselves, we can see that Cry2, take the Cry2 as an example, we can see that Cry2 is highly correlated with the many of the other clock genes, such as the ARNTL, ARNTL2 in the normal samples, denoted in this as a blue dot. But this correlation is largely reduced or lost in the tumor samples, denoted in the red dots. So furthermore, we obtain a data set, the published data set from a published paper. This data is to have a time series data point, and then they have the data in the MCF10A cell line, which is a breast epithelial cell line, also in the MCF7, which is a breast cancer cell line. We identify about 478 genes showed oscillation pattern in the MCF10A cell line, and we can see this is a nice oscillation pattern for those, including those clock genes such as the PER1 and ARNTL. But all these genes lost their oscillation pattern in MCF7, suggesting that there's a disruption of the circadian rhythm in the tumor samples. Interestingly, we convert this analysis, and we can see that there are more than 500 genes showed oscillation pattern in the MCF7 cell line. While comparing with the MCF10A, all these genes lost their oscillation patterns. So this probably suggested that there's a newly established circadian rhythm in cancer. And the interesting question is, what are these genes? So we perform a gene enrichment analysis and found that all these genes kind of enriched in cancer-related signaling pathways, like the TNF signaling pathway and Wnt signaling pathway. So again, these results kind of suggesting that although there's a disruption of the circadian rhythms in cancer, probably there's also a newly established circadian rhythm in cancer, and this newly established circadian rhythm may contribute to the cancer development. Next, because we are doing the cancer research, we always want to know, is there any clinical relevance of those clock genes? So first, we perform a systematic analysis like what we do before. So we identify those clock genes to see whether those genes are associated with the patient's survival. So in this figure, the x-axis is different cancer type, while the y-axis is the clock genes. The red bar indicated that the high expression of these clock genes highly associated with the patient's worse survival, while the blue ones show that the low expression of these clock genes is highly associated with the worst patient survival. We further identify those differentially expressed clock genes among different subtypes, denoted in these yellow bars, while we also identify those clock genes show the differentially expressed pattern among different stages, also denoted in these green bars. So take those survival as an example. So we show that several of the clock genes, such as the CRY2, PER2, PER3, and RRA, their high expression is associated with the better patient survival, suggesting that this gene probably plays as a tumor suppressor gene. In contrast, we also identify many of the clock genes are associated with the worst survival. For example, the NPAS2 and ARNTL2. Their high expression is associated with the worst patient survival. So suggesting that these clock genes probably play as an oncogene in cancer. The next one is we want to know what is the difference among those stages for these clock genes. We identified 23 circadian genes that were differentially expressed among tumor stages. For example, the CRY2 showed a downregulation in the stage 4. More importantly, we want to identify those circadian genes differentially expressed among different cancer subtypes. We know that for tumor subtype information is very important. They provide a crucial insights to help different patients with the different treatment. So for different subtype of patients, there's a need for different treatment. Here we show that for the breast cancer, take the breast cancer as an example. The triple negative breast cancer is kind of have no receptors, and that are the targets of current treatment, and leading the triple negative cancer to a poor prognosis. Here we show that several of the clock genes, such as the ARNTL2, the TNF, they show the upregulation in the triple negative breast cancer. In contrast, we also identify many of the clock genes show the downregulation in the triple negative breast cancer, such as the CRY2 and the PER2. These results suggested that circadian genes probably can be a potential biomarker for particular cancer subtypes. So we have already performed a lot of genomic analysis, but for the cancer treatment, it is always more important to understand the therapeutic liability of these genes. So here we perform a comprehensive analysis to calculate the correlation between the circadian genes and the clinical actionable genes. Clinical actionable genes are those genes targeted by the anti-cancer drugs. So their expression is really important to affect the drug response. So in this figure, the x-axis is the clinical actionable genes, while the y-axis is different color, different of the clock genes, and we can see that the clinical actionable gene actually has a very high correlation with those clock genes. The red ones means the positive correlation between the clock genes and the clinical actionable genes, while the blue dots represent the negative correlation between these clock genes and the circadian clinical actionable genes. We also obtained another data set for the experimental evidence for the protein-protein interaction, and these are marked in the box. So whenever we have a box here, means that there's experimental evidence for the protein-protein interaction between these two genes. We also obtained the ChIP-seq data to show the direct interaction between the clock genes and the clinical actionable genes. So take this as a zoom in. We can see that CRY2 is highly correlated with many of the clinical actionable genes. Like here, they are positively correlated with the gene A11, TSC2. In contrast, they also negatively correlated with many of the clinical actionable genes, such as the JAK3. These correlations is also evidenced by the experimental evidence for protein-protein interactions like this one, and also they have the we have the ChIP-seq evidence to show the direct interactions. Again, the whole figure shows the strong interactions between the COG genes and clinical actionable genes. We obtained another public available data resources. This data generated generated the Cry2 knockdown at the different time point. We can see that those clinical actionable genes show the positive correlation with the Cry2. If we knock down the Cry2, then those clinical actionable genes will show down regulation at different time points. So this is the upper panel. You can see that, for example, the AKT3, they show down regulation when knock down the Cry2. And they also show kind of an oscillation pattern, like this one, the MITF, they show the oscillation pattern, suggesting that the Cry2 is highly correlated or is kind of highly regulated on these clinical actionable genes. In contrast, for those genes negatively correlated with the Cry2, we also observe a similar pattern. So if you knock down Cry2, those clinical actionable genes show the upregulation in most of the time point. Again, we can see the nice oscillation pattern for these clinical actionable genes. These results further suggesting that there's a strong interaction between the COG genes and the clinical actionable genes. Take the EGFR as an example. EGFR plays very important roles in lung cancer. There are many of the drugs designed to target the EGFR, and we can see here there are many of the drugs here. And EGFR is also highly correlated with many of the clock genes here, like positively correlated with the RRA clock EP300, and also negatively correlated with the many of the clock genes, such as the PPP1CA. So when you treat the patients with the EGFR inhibitor, you definitely need to think about or consider about the effects of the circadian, because the circadian, they have the oscillation pattern. Then EGFR itself will have the oscillation pattern. Once we have this direction, we have the high interaction between the COG genes and the clinical actionable genes. Next, we assess the drug response data for those COG genes. So this data we obtained from the GDSC. This data set includes many of the drugs. For example, all listed on the y-axis. These drugs are targeting to the metabolism, histone Wnt signaling pathways. On the x-axis are the COG genes. Again, the red ones indicate the COG genes. The magenta dots represent indicate that there's a positive correlation between the COG gene expression and the drug response, while the green ones indicate that there's a negative correlation between the COG expression and the drug response. And we can see that there are 3 of the drugs have been reported in the previous study. These drugs has been applied in the cancer chronotherapy. So here we show that there are many more drugs show the similar pattern. So when patients are treated with these drugs, they also should be considered as for the cancer chronotherapy. Here we can see that the COG gene and NR1D2 is highly positively correlated with multiple drugs, while the PER2, Cry2 are negatively correlated with many of the drugs. This pattern is largely shaped by the correlation among these COG genes themselves. For example, in the cancer cell lines, the COG genes and Cry1, they have a very high positive correlation, while in the cancer cell line, this Cry2 and PER2, they have high correlation, but Cry2 and this gene COG, they have a quite high negative correlations. So this is the summary for the drug response for these COG genes. We can see that all those COG genes are highly correlated with many of the compounds for their drug response. For example, this PER2, they are negatively correlated with about 40 compounds, and also positively correlated with about 20 compounds. In the contrast, COG is highly correlated with about 60 different drugs. We observed a similar pattern in the CTRP data resources, the Cancer Therapeutic Response Portal. we can see that the PER2 again has a high correlation with many of the drugs here, and also clock has many of the correlation with the drug response. So for those drugs with the known drug targets certain pathways through certain target genes here. So drugs target certain pathways, and through these drug targets, we know that there's a strong correlation or say interactions between these clock genes and drug target genes. Therefore, the clock genes, because they show the oscillation pattern, therefore, if the clock genes show the oscillation pattern at different time point, they definitely will affect the drug response. A recent study published in Science showed that more than 80% of the genes coding for proteins identified as druggable targets by the FDA show the oscillation pattern in transcription in the transcription level, further suggested that it is very important to consider the drug treatment time. Therefore, it is very necessary to develop the cancer chronotherapy. So take the EGFR as an example. We know that several of the drugs, they are targeting EGFR signaling. They are targeting EGFR. We also know that EGFR is highly correlated with many of the clock genes, again, suggesting that if you treat the patient with the EGFR inhibitor, you definitely need to consider about the effects for this oscillation pattern for the EGFR. So to summarize for all this paper, for our paper, we first use the TCGA data resources to show the dysregulation pattern for all those clock genes. We show that more than 90% of the circadian genes show the dysregulation pattern in at least one cancer type. We also show that this dysregulation of this transcriptional dysregulation will lead to the alterations for the signaling pathways. We further showed that these clock genes is highly clinically relevant. They can be associated with the patient survival. They show the up and down in different stages. They show the differential expressed pattern among different tumor subtypes. More importantly, we show that there's a circadian disruption of the circadian pattern in the normal tissues. We also show that probably there's a newly established circadian rhythm pattern, and this probably related to the reprogramming of the circadian rhythm. Finally, we show that there's a strong interactions between the clock genes and their drug targets. Also, these interactions will affect the drug response. Therefore, it is very important to develop the cancer chronotherapy. Always think about when to treat the patient, and you can get the optimal treatment effects. So in the end, I would like to thank the lab member, especially You Chong-ye, who is an outstanding postdoc fellow in my lab. She did almost all the analysis. She drew all those fancy figures. You can see that our figures is really beautiful and very fancy. She drew all those figures, and this is a team science from all help from all the lab members, including Yu Xiang, Zhao Zhang, and also we have a very good collaborators, Dr. Yu from our department, Dr. Takahashi from UT Southwestern. These two faculties are the experts in the circadian field. They give us a lot of advices. Also, Dr. Gordon Mills from MD Anderson, he's a very good expert in the cancer biology, give us a lot of advices on this. So this is really a cross-discipline study, combine the cancer biology and the circadian biology together. So finally, I would like to thank the funding support from the Cancer Prevention and Research Institute of Texas. Without the support, we cannot do this much analysis. And again, if you have any questions, you can send me the email listed here at any time as you wish. I will be very happy to discuss with you. And I just want to stop here, and because this is all the long week, just before the long weekend. Hope everyone have a great long weekend, and I will be happy to take any questions right now. Thank you.
Yes, any question from the audience? It's such a fascinating talk. Thank you, Dr. Han.
Thank you.
Hello.
Yes.
Oh, hi. Thank you for a great talk. I was just wondering how you measured the level of expression of each gene. Did you use quantitative PCR, or what technique was used for that?
You mean the transcriptional regulation in the tumor and the normal samples?
Yeah.
Yeah, that's from TCGA data sources. If you let me show it.
I was just wondering, is it the relative expression level of messenger RNA, or is it something else?
It's a relative expression for the messenger RNA. Actually, the TCGA generated the RNA-seq data. So this is for RNA. This is the RNA-seq for the mRNA expression level.
Okay. Thank you.
Thank you.
Any other question from the audience? I have a question because this is such an insightful talk. I just want to ask from a high level, kind of a 30,000 square feet bird's eye view, what do you think is the current challenge for development of cancer chronotherapy?
Okay, yeah, this is a really very good question. So when we submit this paper, we actually ask about the review as well. That TCGA data, they generate a lot of omics data, but they do not have the time point for those samples. So we do not know is this sample is coming from 2 p.m. or 2 a.m. maybe. So I think for most of the cancer studies, they do not have this kind of time point data for the patient samples. We probably only can obtain a few of the time point data for cancer cell lines, probably mouse models, but it's really difficult to get those time point data for the patients. So recently, there are several of the researchers, they are trying to develop the softwares or algorithms to infer time points from those, based on the expression, to infer the time points when the sample is collected. But still, this is not enough. We know that most of the surgery are kind of occurred during the daytime. Probably no surgeon are willing to go to the patient's room to do the surgery. So the sample for the nighttime is kind of very lacking. I think this is one of the most challenging for the cancer chronotherapy. No data there, so it's kind of limited our analysis a lot. But again, we're trying to improve this, or try to develop this idea so that later on, other physicians, they can try to collect the samples at different time points and provide us more insight for the cancer chronotherapy. Thank you.
That's a great answer. Thank you so much. Other questions from the audience? Okay, I will take this opportunity to ask another one. Sorry, it's just so fascinating. I know that the Cancer Genome Atlas provides multi-omics data resources. So from this data resource, what kind of other information we can get and use for exploration?
Yeah, this is also a great question. So the Cancer Genome Atlas is kind of starting about 10 years ago, and we know that even my former mentor and myself is involved a lot for the TCGA consortium. So again, this TCGA, probably this figure will be better to explain. So TCGA data resources is really a very precious resource for to understand the tumor mechanism for the tumor. So they have these somatic mutations, copy number, gene expression, DNA methylation, micron expression, and RPPA. And the TCGA kind of promote the idea for the pan-cancer analysis. They are trying to compare those sample features across different cancer types. At the beginning, most of the research focus on, for example, the breast cancer, the tissue origin. But now TCGA kind of show that probably there's a more higher level for the molecular alterations for those TCGA, for those cancer patient samples. Back to the 2013, there's a first round of the TCGA pan-can analysis. There are probably 20 or 30 papers published in the Nature series. And more recently, there's a 27 paper published in the cell series. They kind of cover the three large scheme like the cell of the origin, the oncogenic process, and the signaling pathways. They are trying to understand using the TCGA data resources to estimate the immune signatures, stemness scores. They are also kind of mining different kind of elements in the genome, like the enhanced RNAs, long non-coding RNAs. So our previous study also using the TCGA data resources to identify to mining this big data set. Like we quantify the pseudogene expression, linker expression, RNA editing, more recently alternative polyadenation and SNOR expression, eQTR as well. So this is really a very data-rich resources. So I think there's still many things that can be mined from these big data resources. Thank you.
That's wonderful to know. We have this, public database available for all of us to explore in the research community. Yeah, indeed. Any other questions?
Hello, Dr. Han. Can you hear me?
Yes.
Yeah, I have a question. Do you have any data in result in the epigenetic regulation of the clock gene? For example, demethylation in gene promoter region. Yeah, for this.
Yeah, we do have a little about that. We can only see that when you see the overexpression of clock genes, this is likely due to the hypomethylation of the DNA methylation. Yeah, I know you are coming from a lab with the expertise for DNA methylation.
Yeah, thank you. Yeah.
So yeah.
Yeah.
I think there's a lot of regulation for the circadian system. We are more focusing on the transcriptome level, but there's lab they claim there's a kind of alterations or regulation for the DNA methylation, histone methylation. Actually, several of the circadian genes, they are rarely the histone acylation enzyme. They have a histone acylation activity, so they should be very interesting to dig out that.
Okay. Yeah. Thank you. Thank you. Yeah.
So if there's no further questions, let's thank the speaker again. And just don't forget, our next one is on June 13th, 4 p.m. Western time, 7 p.m. Eastern time, about advanced precision medicine through data science. So let's thank the speaker.
Thank you for your patience.
Okay, bye now.
Bye.